Have you told me about that site? You need to reserve that site. Okay, let's see this. What does it show here? Oh, okay, it's not. Somewhere it's missing here. Where is it? Where is it? Where is it? Where is it? Why is it not opening? Wait. Right. So, let's begin. We are testing this as well, right? Is it functioning? No. 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 Or is it this way? So it's got must be on the right. Right. Okay. Does it? Does it not? Good. Foundation, plant requirement. Foundation of plant. Okay. 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 It's alright. We'll no need to log. We're just testing this today. Right. Okay. So let's start. Those who did not come, well, you should know. I will begin when the time is supposed to be beginning with. So welcome to the first lesson for advanced crop physiology for 301. What's your name? Moon. Okay. Undergrad, postgrad? Because this is open for postgrad as well. Okay. All right. Final year? Final year? Okay. Have you started your project?
Not yet, but I'm under your supervision.
Huh?
So for FYP, right, I should be under your supervision.
I do not have any student.
But the list written with your name.
Well, I do not plan to have any student.
Okay.
My advice to you is, I'm having a war with this department, right? Okay, so there's a good chance you're gonna be a victim in the middle. So you need to be smart and shy away from it. You can learn from this class. It's very useful. Yeah, but to be my understanding, you might want to shy away.
So yeah. Does this mean that I need to contact?
Be smart and do not be with me.
Alright.
Do not be with me. It's not like I cannot supervise. I just don't want to. Right? Okay. No, class is fine. You can join because whatever that you learn is going to be relevant for however long science is still around. Okay. So right. So let's start now. So you have registered for this class, right? Okay. So that's that. So I assume that you have taken AGR 43301. That would be the crop physiology, the fundamental one, maybe a year or 2 years back. So this is the continuation for the crop physiology. So it's basically dealing with understanding how the plant functions, so that being in agriculture industry, you can do something to improve the crop production. It has to start with plants first. Okay, because the first concept that you must understand is all crops are plant, but not all plants are crops. So crops are specialized plants which have been highly domesticated, and they have of economic values and importance. So sometimes it can be perspective as well. Some crops, some plants can be crops to certain people, but at certain times the same plants can no longer be regarded as a crop. I'll give you an example. You have a plantation of cornfield. You're growing cornfield. Of course, the plant corn, maize, is your crop because it's a commodity. It's providing you a cash after you have sold the harvest. But suddenly, if you have chili plants in between the rows of your corn crops, do you want to regard the chili plant as a crop now in the middle of the cornfield? It's not, even though for your neighbor who's actively producing tons of chili because she's got a chili sauce business, chili is a crop for her, but chili is a weed for you. You do not want that, right? So it's a perspective. So the terminologies for crops, my point is, can be changed easily based on your interest, based on your perspective. So by learning further into crop physiology, you want to further and profoundly understand what can be done to increase the value of your crops. Because the moment plants do not have any values, they cannot be regarded as crop. It's as simple as that. All right. So is it wrong to say that if you learn plant physiology but you do not do agriculture, it's going to be wasteful? No. If you're just a hobbyist, you enjoy occasional gardening at your home, you will still find the knowledge about plant physiology useful to get some profuse blooming flowers in your garden, to get some prolific produce of your perhaps mangoes, fruits, and all the tropical fruits that you can find easily. So it's very useful. It can cater for your hobby. It can cater for your um, business goal. And also it can cater for many other things that is rather global. For example, carbon sequestration. You see, we are living in a warmer planet now. So high greenhouse gases such as CO2 definitely increasing the temperature of our planet. So by understanding plant physiology, you can do something to the growing plants or forest so that more carbon is uptaken into the body of the plant, so that less carbon in the atmosphere, the temperature can be highly regulated for the whole planet. So that's my point. You learn physiology, you understand it at the cellular level, and you can also have impact at the global level, right? It is special, not like medical, because in medical faculty, they learn physiology as well, but that's human physiology. You learn human physiology, it's true that saves lives, it has impacts on the formulation of drugs, medicine, and so on, but that doesn't really control the climate change, is it? It's good for the humans, but for the globe, for the continuum of the species of other organisms, insects, animals, fish, whales, and so on, human physiology not going to be very helpful. But plant physiology is, right? Because you have some plants or similar looking organism like plants in the ocean that produce lots of oxygen. Right. Okay. I hope I have caught your interest. Why this is a cool thing to learn. So for this particular subject, actually, I'm going to start with quick lesson to refresh your understanding about the fundamental requirements of the plant. And also, in addition to this is also pertaining to your lab. This is not going to be lab work like your regular first year and second year classroom. No, this is meant for highly independent. You need to be in charge, and you need to wear your scientist or investigator hat, although this is not one. So all the scientific conducts that you have learned so far up to this point, you need to put to practice. You need to investigate. So I hope you're familiar with the scientific conduct because you've been doing it for many years now. You have your research question that you want to address, and then you have your objectives, you have hypothesis. So throughout this semester, hopefully we're going to do some small experiments, and I have actually selected some easy to deal with plans so that you don't have to work too much of your time to get some results. Okay, so on that note, for your lab, we are going to to be more focused into it actually. Yeah. So because this is the reality of being a science student. You don't really stay in the class and learn the theory all the time, because trust me, whatever you learn in theory, maybe 60% maximum of it actually happening in the real world. The rest of it, you kind of wonder, this is not what I learned. It's true, because in the reality, life is dynamic, actively changing. This is why textbook got revisions, second edition, third edition, fourth edition, and so on, because the knowledge is actively changing and it's never static. Right. Okay. So for your coming short experiment, we are going to focus on understanding how to improve the plant at cellular level so that it can have effects on the performance of your crop productivity the time you harvest it. Right. So plant, of course, is going to start with the plant cell. Okay, it's got the plant cell. You know what? I'll just use this one because you are all starting here. The plant cell. I hope you should be able to draw some basic plant cells now. It's got the cell wall, got the plasma membrane, and two organelles that we are interested with is, number one is this guy here, and the other one is this guy here. Oops. So this is your chloroplast, and this is your mitochondria. You should know that these two actually will determine the performance of your crop and your harvest. One is producing the sugar, one is producing the energy. So the more you can influence these two to somewhat cooperate, the more of your crop produce that you're going to get. It's at cellular level, but there are many cells in the plant. So of course, if every single cells have been tuned to perform at their best, eventually you're going to get your harvest way more than what you would get if nothing has been done. Right, okay. So these are the two organelles that we're going to learn. And then when it comes to science, there is a concept of manipulated variables. What variables would you want to manipulate to see the effects on your crop? So the variables I'm thinking to play around for the rest of the semester, there's only two. We don't have to go too crazy about it. Number one is, so manipulated variable. Number one is light. Number two is CO2. That's it. Because what? Because light actually will have impact on the light reaction of photosynthesis, and CO2 is actually to feed the Kelvin cycle. And together, this is your photosynthesis. If we are working with regular plants, chili, bean, or anything, it's going to take a very long time. The good thing is we have come up with a system, so we're going to use aquatic plant so that you don't have to take a very long to get your result. So the plant that you're going to use here, which is going to be your constant variable, it's not going to change, is your plant, which is duckweed. Lamina. OSP. Okay, you can have a look at, they are over there. So this is very fast replicating plant. You might wonder, that's something that you would find in the ditch, in the drain. I mean, like, how come that's agriculture? Don't judge too soon. That actually, this plant has a growing interest in the energy capturing technology throughout the world, because it can multiply so fast, meaning that it can capture carbon so much more than any other forest throughout the world. That, and also there are many other cool uses that you can get from this plant. Okay, we don't eat it, but something else will, and those something else are going to be your food, right? I'll give you a quick example of the use. Number one, you can use it as fertilizer. You can use it to fertilize your regular crops. So of course, you eat your crops, right? You eat your vegetables, you eat your fruits, and so on. Then you can also use this if you are carnivorous lover. You can use it as a feedstock. You can use it to feed your chicken, your cattle, and so on. Okay? So there are many uses for this one. And the good thing is, because it's multiplying so fast, meaning that it's going to double in number under 2 days. Every cycle of this, you only need maybe like a week or so. Right? So if you screw up, there's plenty of time to repeat. Hopefully you don't, because this is rather easy. Okay? So, manipulative variables, constant variable. What are the other variables left? Being a scientist, being a science student, you should know. What is it? You should learn about variables, right? How many variables are there? You got constant variables. You got manipulated variables, variables that you change to see the effects. What's the other variables? Okay, responding variables, yeah. So, responding variables, these we can decide later, okay? But quickly, we can see the growth effect of it. Does it get more biomass? Does it get less biomass? And also the photosynthesis to describe chloroplast activity plus respiration to describe the activity of mitochondria. You see, it's all related and connected to each other. So this is another skill that you need to have when you are dealing with science work. You design your experiment. It can be a simple experiment. Science does not have to be super complicated. But whatever that you decide to be your variables must be supporting each other and contributing to the bigger picture of it. Right? So easy. Right. Okay. We're not going to start the experiment today. I just want to give you the overall view about it. And of course, along the way, there are some other small activity that you can do. This is not to cram as much of activity to you as possible. No, at this level, whatever activity in the class or lab that you are going to undertaking, it's going to be hopefully relevant for your coming endeavor. You know, in your working years, in your industrial involvement. You're not going to be in the university forever, right? So I hope that you can make the most of your time while learning this so that you are relevant to the industry and you too become dynamic. Okay, when you are dynamic, you are alive because you're actively changing. If you're not actively changing, what are you? You're dead. Look at the mummy in coffin. Is it actively changing? It's pretty much mummified. It's not going to change anymore. Right. Okay. So we're going to learn more about this in the coming week. So today we'll just quickly. I'm not going to teach too much from this slide. Actually, my focus is actually going to be in these two variables because this is going to be pertaining to what you'll be going to be dealing with. For the rest of the information of the slide, we'll give the video because I've used this slide before for to making the video. Okay, so you can learn for the rest of it. So this is actually the slides that I use for the commoners. You see, in our department, I do training to other people from, not university background, regular people, or people have general interest, or maybe other scientists, but not in the field of plant science, right? So it's good to understand what are the requirements of plants because plants too are living organisms, right? So that's a formula to remember what are the important components to enable plants to grow happily, healthily, and hopefully economically sound for your own interest, right? You can check the Google stream. We have shared with you the slide. So please have it installed in your phone because that's our means of communication. Okay, if you missed it, well, that's too bad. Right. So 4 components that will determine whether the plants are going to live or otherwise: air, light, water, nutrient, and growth manners. Meaning to say, if you fulfill these fundamental requirements for the plants, you can pretty much grow the plants anywhere, even in a mouse, in the cave, in the Milky Way galaxy far away, in some kind of random nebula. Doesn't matter. As long as the plants receive all of this, they don't care. Wherever is the location they are growing in, right? Meaning plants do not require gravity. Many scientists have done growing plants in the International Space Station. Well, the plants are growing, even though they look funky when they are growing, but they're still growing and producing all the flowers as usual. So they have decided that and concluded gravity is not essential for the growth of the plant. It can be essential for us because without gravity, many astronauts in the space station, after they have spent quite some time in the space, when they return to Earth, their bone density got greatly decreased. So they become osteoporosis at a very early age. So gravity is actually what makes us human. You know, we can walk on our 2 feet. Look at around other mammals. Can you see any other example of other mammals, animals walking and running and doing all sort of activities on 2 feet? Or your cat might, if it's trying to fight you back. You know, some cat can walk on 2 feet. Think about it. No, they have to be on the falls, right? So that what makes us human. So because of gravity, that really shaped the forms of our bones and also the density of our bones. All right. So we are actually, when we are on the sea level, actively against the gravity, and that actually what strengthened our bones. So when there's no gravity, yeah, your bones kind of give up. I mean, what? That's it. I'll just don't grow any more bones or repair. So that will be the end of you. You basically turn into a jellyfish very soon. Right. So air. Let's see. Air. There are many components of air that are of importance to the plant. Oxygen, of course, and humidity. Humidity is the amount of moisture that is present in the air, and also the temperature, which is basically how hot or how cold it is, and also the CO2 concentration. So we are going to focus on this guy, CO2, because that's going to be related to your experiment. But I will mention quickly about other things. So temperature. Why temperature is important for plants? Simply because your plants are living organisms, and living organisms, they are full of proteins. You too are full of proteins. What happens to protein when it gets too hot? It gets denatured. Can the protein function when it's denatured? Pretty much you cannot. Yeah. You have your egg. You just bought your egg from Jaya Grocer. Wait, your egg is fine. You crack your egg. It's still in liquid form. The moment you boil your eggs, it becomes solid, right? Can you reverse the solid egg becomes the liquidy egg? It does not serve back the function. Therefore, the boiled eggs cannot produce chicken, no matter how much you cry. It's going to stay boiled egg forever. So the same concept applies to plant. In a plant, there are so many vital proteins that are actually temperature sensitive. Once you have hit a certain threshold, especially the common inhibitory temperature around 42 degree, these proteins can no longer function and they can't repair themselves, and that will be the end of it. Right? So very important temperature. And I've shown there are some images of the plants as you increase the temperature. Of course, if it's too cold, it's not going to do the plant any good too, because temperature, the heat, is actually energy. Do you know tundra? Tundra in the poles, the North Pole? You compare the tundra ecosystem and our tropical ecosystem. Which ecosystem is teeming with activity? Super busy, so many diversity. Tropical, right? What about the tundra? Does it look as busy? Does it look as noisy? So many tigers, cat chasing around an elephant? No, because there's no energy there. There's no energy. So rather dormant, rather quiet. But as it gets warmer, as in our tropical ecosystem, you can see that it's teeming with life activities. You know, you just want to shut up all the monkeys. You know, making the sounds, the birds chirping non-stop. right? But if you further increase the temperature, now we go to Sahara. Is it still noisy? Now it becomes quiet, just as quiet as the tundra. So that's kind of the analogy here. So we are in what the ecosystem scientists call it. We are in Goldilocks zone. Have you heard that before? Goldilocks zone. Goldilocks zone, meaning that it's a sweet spot. Sweet spot. Not too cold, not too hot. Our Earth is actually in the Goldilocks zone in the universe. If it's too hot, like Venus and Mercury planet, pretty much nothing can happen. Two, if it's a bit too far, like Mars, Jupiter, and Saturn, the planets, it's too cold, nothing much going on as well. So our planet too is in the Goldilocks zone in the solar system. So don't you find being in Malaysia is actually rather fortunate and a happy place? Not only that you are in the Goldilocks zoned planet, you are also in the Goldilocks zoned ecosystem. Double, triple the fortune. Right, right. What do you think? All the colonizer came here to colonize the land. Yeah, yeah. So be grateful of. I know there are many imperfections here and there, but well, you two are not perfect. So why do you expect everything to be perfect? Right, right. Okay, and then humidity. Humidity is basically the amount of water vapor that is present in the air. You can't see it. You need to understand this one concept easily. Not only gas, oxygen, nitrogen, carbon dioxide that is present, water, gas also present in this vicinity that you are living. So the important thing to take home here is, if the humidity is too low, meaning it's rather dry, the stomata of the plants are going to close. When stomata close, there is no gas exchange, the plant cannot photosynthesize well. That's it. Usually the RH below 50%. But if the RH is too high, above 85% or 90%, near saturation, the plants are going to be attacked by fungal growth. Okay, so it's important for the plant to live in the, again, Goldilocks zone of relative humidity, so that the stomata can open just nice to facilitate gas exchange. All right. Oxygen, of course, we need oxygen, you need oxygen, plant need oxygen, and that is actually one of the important ingredients when we are dealing with mitochondria, cellular respiration. Okay, without oxygen, you cannot do your anaerobic respiration. Plants cannot do aerobic respiration too. So do not think that plants do not require oxygen. It's true, plants produce oxygen as a byproduct of photosynthesis, but plants also use oxygen for their mitochondrial activity, and especially at night. Okay, but too much oxygen, you will learn later, too much oxygen, not a good thing for plants. It will hamper photosynthesis by the process called photorespiration. Okay, and CO2 concentration. CO2, that will be one of your manipulated variable later, the CO2. So, what do you need to understand about CO2? CO2, one of the ingredients of photosynthesis. As simple as that. Why? I do not know. Ask the evolution. It's the decision made millions of years ago. Somebody might ask, why the plants do not use nitrogen as ingredient for photosynthesis? It's hard to answer that question, right? Yeah, but it's good to know CO2 is actually our waste product. If our waste product is not consumed by other organism, we are going to live in our own filth all the time. So thanks to plant, that is good being cleared out. So What's important about CO2? Carbon dioxide is one of the greenhouse gases. That's number one. There are 7 of them. You can find out later. Nitrous oxide, sulfur dioxide, and so on. It's the concentration of CO2 in the atmosphere will determine crop productivity globally. Okay. In the past, I'm talking about before the industrial revolution, way before. Let's go back to Jesus' time. CO2, like a quarter of what's now, or less than that actually, around 150 ppm part per million. Okay, so good for Jesus. There's no global warming, but you don't expect the plants to flourish so much because of this low CO2, which is fine because compared to Jesus' time and today's time, the population is much lesser back then. So less fruits and vegetables are required to satisfy all the following apostles. But with the I'm thinking of a word, so new word so that you can to learn. Anthropologic activities. Have you heard that before? Anthropologic. Oops. I'm not a linguistic guy. Anthropology. Human activities. I might. Can you check the spelling? Anthropology. Meaning that human activities such as what? Timber, digging for the oil and so on. All the human activities. All these anthropological activities, such as happening in the Industrial Revolution. Really, is that right? Anthropology. I know it's right. Hardly I got the spelling wrong. Otherwise, I would have won the Scrabble. With all the activities due to human interest, this has greatly increased the CO2. Okay, just now I said high CO2 means more plant production. So, and now this is what happened during the Industrial Revolution, right? High CO2 released into the atmosphere, and now from 100 ppm or below 100 ppm, all the way now the CO2 is 425. Or is it? Let's see. I will drop the link to the chat so that you can check yourself. This is why you take a course like this so that you know. CO2. For our planet, there is a observatory to check this CO2. It's not in Jerantuk Pahang, no. It's actually at the top of volcanic mountain, free from any contamination, because this reading represent the whole of the planet. I'll drop it to your. All right, you got it. GML.NOA. That's actually the Global Monitoring Laboratory. And they got this observatory in Mauna Loa, somewhere in Hawaii, far away from anthropologic activities, far away from any unnecessary high release of CO2. So if you scroll down, you can see that the CO2 now is, I said just now 425, right? What's today? February 26th. 429. Yeah. 425 was actually, I checked in January. And guess what? During my student time, it was 395. That was like 20 years ago. 395. So it's increasing. So it's real. 395 jumped all the way to 429, How much percent increase is that? Anybody good with mathematics? 395 jump to 429. How much percentage increases that? 10? 10% in under 20 years. Jesus' time to industrial revolution, that's 2000 years. 2000 years increase about 200 ppm. That's a much longer time span. And now only under 20 years it increased 40, like what, 30 ppm. So what's going to happen in 10 years time, a decade from now? It's going to be good for your plants, but is it going to be good for you? This is why you learn agriculture with the correct science, so that when you do agricultural activity, it's not only going to satisfy your crop production needs, for your profit, for your food, but you must have a respect to the environment. You need to make it sustainable. You don't want, in the process of getting all your fruit produce, you are actually burning the whole planet. I mean, like, the one who's going to be at loss soon will be we ourselves. Okay, so this is what learning plant physiology is all about. One of the many reasons. Increasing production is one thing, but also do it in a rather sustainable way. If we go at the pace that we are going now, our planet will turn into moon in no time. With craters and everything. Well, there are craters now happening. So yeah, that's not good. All right. Okay. So therefore, we understand with some experiment later, CO2 is good, but what happens if it's too much? If you increase the CO2 by 20%, can you get the increase in biomass by 20%? Or is it not proportionately going up? Or is there some kind of a terminology? There's a concept of too much of good things. Too much of good things. You think it's happy having a partner, boyfriend and girlfriend, too happy. So you have 10 of them. Do you think that's going to be happy? What will happen? One is, of course, happy. Try 10. Why 10? Morning different, night different. We take turns. Too much of a good thing. What's going to happen to you after 1 month? Number one, your ATM card will turn negative. That's for sure. All right. Okay. So we're going to see what's going to happen if it's too much of CO2. That's why we cannot allow this planet to acquire and piling up more CO2. 429 is worrying already. I think you know why 429 is increasing sudden. There's a sudden jump. You know why? Number one is because actually there are more land mass in the northern hemisphere compared to southern hemisphere. We are on the equator, right? So much of the land is actually on the northern hemisphere. In the northern hemisphere now, we have entered the spring and approaching summer. So these are the times of photosynthesizing for the plants and also other activities and so on. Not only plants, but also all other animals. Animals that was once sleeping, all the bears are awakened now to terrorize the village. It's spring. Let's make babies. So to top it off, what's going on in the Middle East now? Do you not think something get released every time something is fired? That's a lot. That's a lot. Okay? A lot. All of these CO2 have impact and you can see it in real time. Right? Okay? So again, firing everything also under here. Not firing your water pistol. This is the real thing. Right? Okay. So we learn about CO2. Right. Okay. The other one, the final one is about light. Light is actually a very long story. I'll try to compress it so we can be done in 15 minutes. Do you like physics? Did you take physics during your yeah? How's your wave optics physics? Did you enjoy it? Have you heard the word quantum physics before? Did you enjoy learning that? Or no idea what's going on? You see, many of the agriculture people, one thing that they do not do is to learn properly about light, because they just leave it to the physicist to learn about light. Actually, that's not quite correct, because light is one of the ingredients for photosynthesis. Therefore, even though it's hard to grasp the whole concept of light, because it's a mystery, I'll tell you in a bit why it's a mystery. It has to be understood so that it can be manipulated to give impact to your interest. Okay? So we're going to light, which is important for agriculture. You need to understand there are 4 dimensions now: spectrum, intensity, photoperiod, and also angle. Well, before this, I didn't put the angle. Now I put the angle. Spectrum dealing with the color, intensity dealing with the, is it bright, is it dim, or is it dark, right? Photoperiod is basically how long is the light for, 2 hours, 6 hours, 12 hours, 24 hours. And angle is the from which direction the light is coming, all right? Is it coming from, of course, it's coming from the sunrise, yeah, and so on, right? So we're going to look at this light as a mystery, because even without all these 4 dimensions important for photosynthesis, you're going to find out that by learning light, you know that, oh, this guy is very enigmatic. Oh, that's a new word for you to learn. You know enigma? That's actually a machine used in Nazi World War. Enigma. It means mystery. And when I say enigmatic, meaning it's a mystery. Why is it mystery? It has what we call as the dual nature of light. Dual nature of light. So does it mean that the light is a training? No, it's not hermaphrodite. Or is it? Meaning that it has 2 properties at the same time. How come? Light can come as a form of particle. Light can also come in the form of wave. When light comes as a form of particle, you call it photon. Therefore, you can count how many lights coming your way. Imagine you can count the light. Yes, there are instruments that can count the photon particles of light. But if the light comes in the form of wave, meaning that it carries energy to you, like the sound wave energy. You know, if I bang here, you can hear all the way to over there. I'm speaking to you, you can hear all the way back to the class. That's sound wave. This is light wave. So with this nature, they come together and you decide which properties you want to pay attention to. Okay? If you want to pay attention at the time, you want to pay attention to the particle, that means you are dealing with intensity of light. Intensity of light. So intensity of light meaning that is it dim, is it dark, or is it bright? So if you use your phone, you can actually quantify it. There are many. Can you share to the class the GPS status? If you can open, there's a apps. Did you know that your phone got a light sensor? So you can use an app to turn on that light sensor, and then you can take the light reading. So if you can download to your phone and open that, it's called, I just reinstalled this thing actually, it didn't work earlier. It's called, where's that GPS status? Oh, it's out there. Yep. So I'll take the light reading here. So maybe now, just over here. I'll take one here. So you can see that the light there is, where's the chat? Photos. This is why you cannot be IT illiterate. So that's the light. I just shared in the group the screenshot of the light. You can look at the bottom right. The brightness, the unit is lux. So that's around 568. It's just here. Now let's try right under this growing chamber. So, whoops. Right. And I'll add another one. So what's the reading under there? How much lux is it? 2000, right? 2000. To your eyes, I mean, like, okay, that's bright. This is also bright. I can read under this light. I can read under that light. It's nothing wrong. Nothing wrong with it. Nothing wrong with it. Okay. But in the eyes of the plants, this does matter because light is energy, and light also have color that the plants will use at different times for different purpose. Okay. So these lux do not tell you about the color of the light, do not tell you about the duration of light, do not tell you about the angle of light. It only tells you only one dimension of light, which is intensity. That's it. This is why I said with light, you need to talk about one dimension at one time, even though actually there are like 4 things coming at the same time, right? Okay, and then the light spectrum, it's all right. That's actually the color, okay? Coincidentally, the color of rainbow is also the color can be perceived by your eyes retina. If you look at the slide, there is a, can you see the eye cross section there? So at the back of your eye, there is a tissue called retina, and these retina are embedded with light photoreceptors. You know, photoreceptors are specialized cells to perceive only specific signals. So there are cone cells and also the rod cells. So these cells, they can perceive specifically the wavelength of light from 400 nanometer to 700 nanometer. And guess what? This is also the range of your rainbow. 400 is the start of it, which is the blue. And you got all the color in between. And finally, you got your red. Do you remember all the colors in rainbow in sequence? What? You start with what? Red. Red means it's 700 nanometer, meaning that is the lowest energy. Lowest energy. After red, what? What? You need to wake up. Why don't you write the color? It has to be in sequence. It cannot be just as you play. You can write one friend. I know you just met your friend. Just pretend you have known your friend forever. You can check the sequence. The sequence cannot be changed. Okay, this is fixed because it's corresponding to specific energy. Is it right? Is it right? Okay, good. Thank you. So, and the lowest one is actually 400 nanometers, and this is the highest energy. Okay, this is the longest wavelength. It's opposite correlation. Okay, you need to get this right. Longest wavelength, lowest energy. Shortest wavelength, highest energy. This is lambda. Lambda is the universal sign for wavelength. Okay, and what is the lambda here? 700 nanometer. What's the lambda here? 400 nanometer. It's the opposite, right? So if you look at the scale on your screen, all the way to the left, you can see that ultraviolet. Why is it black? Because your eyes cannot see it anymore. It's there, but you can't see it. All right? So what's the appreciation that you can get from here? You see that there are two bars of light there. So there is the rainbow bar. I want you to look at the first bar at the top. The one with the most of it is black. Just a small strip with the rainbow. You see the black bar? That is the universe. The amount of light throughout the cosmic that nobody can see. So next time when you look at the sky at night, you see two stars. Rest assured that the two stars that you can see are giving out the light of this rainbow. But in between two stars, definitely there is something, but you can't see because the wavelength is in the black region. Before 400 nanometers or beyond 400 nanometers. It's actually beyond 700 nanometers. If it's before, it's going to burn you, fry you up. Right? Okay? So, when you learn plant physiology, you need to learn about light because it's an ingredient or input of agriculture. So, how this relates to your experiment? So, you're going to grow your duckweed in two locations. One is under there. That's the artificial light. You can see, can you see the light strip there? It's got, what color? What color can you see? Or can you not see? What color? Yellow. You need to tell me if you are color compromised with your eyes. Because I got a student with color compromise. He keeps calling everything yellow. What color can you see? Green? Is there any red there? Is there any red there? Why don't you come here and say, look at? Is it the only? Come. Because all these light tubes, they are of different LED combination. Come and have a look. Come, have a look 1 or 2 minutes. Then tell me what you see. It's okay. That's mine. I built that. It's not somebody else's. Not all light tubes are the same. How many lights are there? The front light tube, the back light tube. How many lights can you see? And you can test your apps now because you got it in your hand, right? Don't be shy. You want to be a science student? Don't be shy. You be shy. You go to hole, and nobody going to come and save you. Definitely not me. At a regular distance from, maybe you see 3 kind of light tubes. See at the same distance, are they of the same lux value? You've got different tiers, right? The top tier and also the bottom tiers. Is it turned on, the bottom tiers? And of course, at the back, you can see the white light. Okay, all right. Okay, come back. How many lights can you see? How many different light tubes are there? 4. Okay. What light tubes do you have? Red, blue, yellow. Yellow. Interesting. Yellow. And then white. Is it really white? Is it really white? I'll take this. Oh. Let's see this thing. You see that? Oops. You see that is 40. Why is it not written here? This one is eco-link. Means output 4000K. For some reason, it's not written here. That's the voltage. Oh, it doesn't. It use the lumen. I don't want lumen. Anyway, right. So the red that you see. My question now is, there are different light tubes. Are they of the same red? 700 nanometer. This is all. Why not? No ink. Throw it away. These are all red. These are all different shades of red. They're all red. So my question is, do you see any difference Right in there. This is all about being a good science student. You can detect the difference while others cannot. Oh, blue is blue. No. You are dealing with the spectrum now. What kind of red? Are they all the same? Reds? Are they all the same? Are they all the same? Can you see the yellow one? The yellow one is actually having this five, I think 550 something. So it's got this region, and the red that you see is actually this. It's a combination of these two. So that's why it looks rather yellowish. It's a combination of energy. Okay. Or if you will see later, there are many different units to quantify light spectrum like this. But for now, you have learned so far, you can quantify light, the color. Besides color, there's a synonym for the color of light. Meaning that if I say 700 nanometer of light, that's actually synonym to I want red light. You just learn a second language in quantum physics. I just want you to realize that. So when you go to IKEA shopping with your fourth partner night, like babe, what do you want to buy? 680 nanometer light, please. Do you want to use that language and go to the IKEA cashier? Yes, ma'am. What would you like? 680 nanometer light bulb, please. Is it wrong? But is it sensible? So when you say the red light, she immediately understand. So being a scientist, this is my point. Open up your horizon. There are many things talking about the same things, but it's got its own language. When you understand this language, you can do your own investigation to get your scientific endeavor properly done. Yeah. Sometimes when you Google look for things, you're not finding anything because you do not put the right keywords. Now that you have learned, good for you. All right. Okay. And finally, it's the intensity. Intensity is just the number of photons per given area. So usually, okay, there's an example there. You have a light source at the top. The closer you are, just now you are playing with your phone, right? When you are closer to the light source, the lux get more, 1,000, 3,000. As you go farther, it becomes under 1,000. So that's the concept here. Why? Because the spread of the photons get diluted. Because it's basically particles. It's basically particles. So particles have the properties of to be dense or to be very, very scarce. Okay? And of course, when you're talking about the particles number, don't talk about the color. One thing at a time. One thing at a time. Right? And of course, finally is the light photo period, which is this is not a problem to Malaysia as well. It is important if we live in a four-season country or temperate. Many of our plants in our tropical regions, they are day neutral, meaning that they do not require specific length of light in order to flower or in order to bloom. Yeah. But if you go to Cameron Highland, there are many people growing flowers, right? You can see that at night they turn on the light. Okay, that actually for the chrysanthemum because the order is not in yet. So they prolong the light so that the chrysanthemums stay in vegetative phase, not blooming. Okay, and then there's suddenly next morning there is a landslide, a bunch of amar died. Okay, now orders coming in, we need flowers for the funeral. So they turn off the light, the plants bloom, and the funeral is next week. So just enough time to fulfill the order. Right? Okay? All right. Okay, that's all. That's all for today. The rest of it, don't worry. I just want to focus on these two parameter. All right. Okay. Any question? Good? 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 Okay. So next week, when's your lecture? When?
Tuesday.
Tuesday. What a time? Morning. Okay. So in that case, I'll see you during lecture time. You can just come here. Just come here. I hope we can start doing this during your class time. Remember, like I said, this is not about learning the theory so much now. It's more into practical. From time to time, I'll give you a short lecture like this, but the rest of it is for you to put into practice. You do quick experiment because all this while you are like the photocopying machine. You just memorizing, and most of the time you don't even know what you are memorizing, and you got the information mixed out with each other. So let's work with your hands. Work with your hand. And quick disclaimer: I'm not going to give you any lab handouts. So my verbal explanation, instruction, it's your responsibility to create your own lab handout. You need to be a responsible scientist. Please do not think you are first year who feel entitled to be spoon-fed. That's not healthy. Okay? It's not like I'm going too fast. Am I going too fast? Relax, right? Relax. Okay? So hopefully next week we will set up the cups for you to grow. We will grow in the light here, the LED light, and also next window, that window. Right. So have you taken your statistics? Okay. So I will ask, are you going to, which design are you going to use? RCBD, CRD, Latin square? Which one you want to use? And I'm not going to tell that. Look at the situation. You need to make your own judgment what design to use. Right. So the good thing is, this thing is very fast, right? If you got it wrong, you got time. Okay? So we're going to start with this first. The light and into location. So if that was ANOVA, how many factors are there? Have you learned your ANOVA? So we got light here and here, and using this. So you have until Tuesday to think about it. Because if you get it wrong, your experimental design will be erratic and you will analyze your data wrongly. Statistics are important, but it has to start with correct design first. Or maybe you cannot use CRD. Oh, I have to use a nested design for it. Oh, that's fine. Use it. You learn it. You learn it right. Now let's see how far your A will take you. I'm not going to teach you that because I'm not a biostatistician. I can only tell that's wrong, that's right. That's all I can tell. All right? Okay? Good? All right. Okay. So no question. I'll see you next Tuesday here. All right? What time? 2. All right. Okay. I'll see you later.